Hey everyone, welcome to review time. So we did a couple of FRQs over the last couple of days, and then I started looking through the curriculum for the AP Biology test, and I decided to put together this PowerPoint. It's probably going to end up being hundreds of slides, and each section I'll make a video for. So here we are. We are in the first section. And the first section is going to deal with a little bit of chemistry and some of the review that we did earlier in the year. So let's see what we got. All right. So Unit 1 is known as the chemistry of life. And that's basically what it's going to be. A little bit of chemistry, a little bit of pretty much water, macromolecules, some carbon, organic things. So let's see where we're at. As you can see, I start each topic with a something, 1.1, 3.2, whatever. And that's the major topic. And then underneath that is kind of the big idea of the topic. So here we're looking at water and the structure of it. And living systems are organized in a hierarchy of structural levels that interact. So that's what we're going to see, building on molecules, breaking of molecules, and water is a major player here. So each of the subsections has kind of like a big idea. So what I did was I turned it into a question. And a lot of you sent me messages saying, well, why are you making us do this if the answers are on the next few slides? Well, what I want you to do is see what you remember. So the question comes and then the answers. What I do not want you to do is go look at all the answers and then write it down. That defeats the purpose. What I want you to do is see what you remember. And this part here is not going to be graded for a grade. It's going to be graded for completeness. The FRQs at the end and the videos that you write about, they're going to be graded. So here it says, what are the subcomponents of the water molecule and how do they determine the properties of this molecule? So I was looking for something along the lines of it's got one oxygen and it's got two hydrogens. And something along the lines of that oxygen is extremely electronegative. And what that means is it kind of likes to hog the electrons, but it makes a covalent bond. So for example, you have your oxygen, you have a hydrogen, you have another hydrogen, there's your H2O. Oxygen has what is known as a partial negative charge, which leaves the hydrogens to have a partial positive. And it's not a true positive or negative like it would be in an ion situation. It's just that the oxygen is such a hog that it's kind of pulling the electrons that they're sharing closer to it because it's like a little magnetic hog, and it kind of gets a little bit of a negative quality to it. That's what's going to allow for this hydrogen bonding that we'll see in a few seconds. So if you answered something like that, you're good to go. So polarity is the key here. Water is a polar molecule, and it has what are known as polar covalent bonds. So they're covalent because they're sharing the electron, but they're polar in that one is kind of a little bit more greedy. So the way I drew it before is instead of the electrons being shared nice and neatly in between, they're closer to the oxygen, which gives it that partial negative. So let's see what happens. The reason why this polar covalent bond is important is because, once again, it sets up this kind of polarity situation. Now, a hydrogen bond is not a real true bond where there's any kind of sharing of electrons. It's more of a force. But you can see it here that these water molecules are held together to each other, which is kind of cool. And that is what is making this thing called a hydrogen bond. So there's a really good chance that one of the FRQs you're going to get is going to have to do with hydrogen bonding and or water's ability to play in the making and breaking of macromolecules, which again we're going to see coming up later. So once again, the negative of the oxygen, the positive of the hydrogen, it causes these things called a hydrogen bond. This is going to go on to be something called cohesion, which we will see in a second. But basically, that's a water to a water in this case. But we'll see it coming up in a second. So in both of these pictures, you can see here that these dotted lines represent hydrogen bonds right there. They didn't even bother putting the bond here between the hydrogen and the oxygen. No, they kind of put it right there, but they didn't really go too crazy on this diagram. You can see they just kind of butted them next to each other. Once again, that is a covalent bond, a special covalent bond called a polar covalent bond. These guys here are attractions. They can be broken by adding a lot of heat. That's evaporation. All right, so listen, describe how living systems depend on the properties of water that result from its polarity. Once again, if you said something along the lines of cohesion, that's how water molecules stick to each other, which is going to ultimately help lead to something called surface tension. If you said something along the lines of that, I'm happy. If you said something along the lines of that water has a high specific heat, so therefore it's hard to kind of get these water molecules apart, so it takes a tremendous amount of energy to heat up and to cool off water. If you said anything along the lines of that, all good. If you said anything along the lines of water is less dense when it freezes, so it floats, so therefore fish can kind of still swim or survive in the water underneath it. So if you said anything along those lines, once again, you did a good job. So let's talk about a couple of them. Stabilizing temperature. You guys take this for granted. You don't even think about it. But what happens is if you have some land, let's say even Long Island, and it's butted up against the ocean, there is a much different climate right here on the land than there is very far inland. So places like the middle of Pennsylvania, let's just say, they're on the same latitude as Long Island, but their temperature ranges are very, very, very different, and that's going to allow for different kind of plants. It's going to allow for different planting seasons. It's going to allow for a lot of different things. So water stabilizes temperatures, and that is because of what is known as its very high specific heat. It means it takes a lot of energy to heat and to cool water. And that keeps, again, our temperature range from getting too crazy. So around here, it very rarely gets up to be over 100 degrees, very rarely, and it very rarely gets to be negative or even negative numbers Fahrenheit, very rarely. Water is an excellent solvent. This is going to play big time in the world of erosion and the ability and weathering of water to kind of break down things. Now, where we see a big deal is salt, and this is what happens in the oceans. So that's, again, a pretty big deal. A lot of organisms depend on that salty water for their osmoregulation, which we'll talk about later. And then, of course, cohesion or cohesive forces is a water... darn it. Let's see if this will erase. So it does. Is a water to a water. Adhesion is a water to some... I don't know why it keeps on doing that. A water to something else. And we'll see what that something else is in a little bit. And that is going to lead to surface tension. Okay. So stabilizing temperatures. Let's go right on out here. This is, looks like California. Yep. And you can see here that places like Half Moon Bay that are right on the Pacific. The temperature that day is 67. Going inland to Livermore is not that many miles. And sometimes you'll hear it referred to as the valley if you watch enough of these TV shows. But, I mean, that's like almost 40 degrees temperature difference. No joke. And that's going to allow for many different plants and animals to survive or not survive, depending on that temperature range. The fact that it's an excellent solvent. If you look here, when sodium chloride, which again is your regular salt, when you mix it in water, what happens is it basically, the ionic bond breaks, and you can see here that they separate. And when they separate, the chlorine has a negative charge and the sodium has a positive charge. Well, water's like, okay, we can be friends. And if you notice here, the positive hydrogens are kind of linking to the negative chlorine. And here, the positive sodium and the negative oxygen are happy. So this is just a way to break it down or dissolve it. So kind of cool how that works. Cohesion, adhesion, and surface tension, we kind of mentioned them all already, but if you said something along the lines of cohesion is a water molecule to a water molecule, that's great. If you said adhesion is a water molecule to something else, and once again, that something else can be a lot of things, and that could lead to something called water tension. So for example, let's say we have, I don't know, a blade of grass, let's just say right there, and you may have seen this in dew in the morning, and there may be a big old droplet of water right on the end of that piece of grass. What holds the water to the piece of grass, there's your adhesion, and what holds the water molecules together to kind of make this bubble, that's your cohesion. So we'll see a little bit more about that when we talk about bugs that can literally walk on water. So cohesion and adhesion, here they are again. Cohesion, they kind of kept it this way. That's a water to a water. Adhesion is a water. Once again, there's something else. I've now said this a bunch of times. And it has to be a polar substance. Water does not stick to things that are non-polar. So this has to be a polar substance. So if something was super greasy, that would not work, because things that are greasy or oily are non-polar. But again, we'll talk about those later as we go on. So here's the surface tension, and this is kind of a fun diagram. I'm not sure you've seen a lot. Something called the water strider or many other types of bugs have the ability to walk on water, because if you look here, their feet they're spread out so much and they're so light that they don't break the surface tension. And what we did, when we tried to float the paperclip on the surface of the water when we did that in the lab. Sometimes you'll also see, let's say, a beaker of water or a cup of water where it appears that the actual water in it's kind of bubbling up and over. That's your surface tension. And that could be helpful for animals like creatures that walk on the water. How does transpiration work? Now, this is a big deal. Unfortunately, we didn't really get to finish the whole photosynthesis thing because of the breakup of the school year. But what happens is, and I kind of drew this out and I talked about it, but we didn't really actually do it. If this is a plant's going to have some roots, and this is going to be the ground. So what's going to happen is it's going to rain, or you're going to water the ground or whatever. Now the water is going to sink in, and we'll talk about the whole water potential thing and the whole way it moves later. But what is ultimately going to happen is the water is going to move into the roots. Now, once it gets into the roots, there's going to be vessels, or they call it vascular tissue. So I'm going to kind of draw it a little bit big here. This is going to be a vascular tissue called xylem. Now, xylem is we'll call them a series of tubes inside of a plant that are going to carry the water up. Now, the key here, which is kind of important to understand, and I don't really know how much you knew about this, but water is going to evaporate. So when water evaporates, it's going to evaporate because of the sun, it's warm, whatever. Because water is polar, it's going to be attracted to its friends, its other water molecules. So let's think about them being linked in as a train. So as one water molecule kind of gets released off out into the atmosphere, it pulls up its buddies because of these cohesive properties. And at the same time, these xylem are very, very, very thin tubes. And what will happen is the water will sort of kind of cling to the side of that. That's the adhesion portion. So the combination of adhesion and cohesion working together will draw water up through a plant, through the leaves, and out. Again, if you do not water a plant, eventually it will die. So that's the kind of finish up the transpiration thing. Now, this picture here is kind of cool. Same thing, fancier version, of course. There's the water going into the roots. You can see here that there's your soil, there's your water. You could see it entering, and that's a whole water potential thing. But again, we'll do that later. Then, if you see the zoom in here, you can see the walls of the xylem, and you can see each individual water molecule kind of like a train, kind of hanging onto each other, and along the way sticking to the walls of the xylem. So again, pretty neat. And eventually leaving out the leaves through evaporation. So that's transpiration. Again, pretty important. You may see that again at some point. A couple of videos some people were asking. So when you click on them, I tried to pick videos that are somewhere between about 8 and 12 or 8 and 13 minutes, something like that. And what I want you to do is just watch them and create questions from the videos. And typically, if you watch a video as from a teacher's point of view, you may be more likely to actually pay attention to what you're doing. So you'll submit those on a separate document. So there's the first two. I believe this section had 3. Now here's the FRQ. Now what's going to happen is this will get graded. So there's going to be a lot of right answers, and I'll go over them as we talk about this here. But let's look at, for example, choice A. Describe 3 properties of water. Now, the 3 properties of water you could have said were, we're going to come back to this, you could have said something along the lines of cohesion. You could have said something along the words adhesion. You could have said it's a good solvent. You could have said surface tension. You could have said stabilizes temperature. Now, what I'm going to try to do is make these FRQs answerable from the notes before it. So as long as you've been paying attention and you kind of go through the notes and you read them and you kind of look at the answers, the FRQs should be pretty easy. What I want you to start to do here is learn the basics and what to do and what not to do. So let's kind of go back and I'll show you what I'm talking about. So you just describe it. So if you said something along the lines of, cohesion is water to water, or water being attracted to another water molecule. Adhesion is water being attracted to some other polar substance. The fact that it's a good solvent, it can break up ionic bonds and things like salt. Surface tension, it allows for water to almost form droplets so they can form as rain, fall as rain, or they kind of create this surface on a lake that allows animals to walk on it. Stabilizing temperature, if you said something along the lines of high specific heat and cool slow, regulates the temperature of the climate, that would be perfect. So you hit each point. Now going to B, explain how the properties of water are used to regulate temperatures within living things and the environment, which again, that's going to focus on the high specific heat, and that's also going to keep plants that are wet cool, because if a plant has a lot of water in it, then it will also heat and cool very slowly. The movement of water through plants, which we talked about in transpiration, and its involvement in cell respiration and photosynthesis. Now, we did not talk about that, so that's what I'm going to kind of focus on here. In respiration, water is formed because if you remember, oxygen was the final electron acceptor, and when those hydrogens came screaming through the electron transport chain, water was formed. So in the world of respiration, which again we'll talk more about in chapter 3, but that's the formation of water. And in photosynthesis, which we just finished up, water that is brought into photosynthesis is broken up, where the hydrogens are going to go one way and the oxygens that are kind of produced through multiple waters, they are going to be released as a gas into the atmosphere. So if you said something along the lines of all that, you're perfect. All right. So that's that question. That's that question. All right, 1.2. This is the elements of life. So what are we going to need? What are some of the elements we're going to need? Well, the things we talked about. Well, things like carbon. That's a huge one. Carbon's going to be found in all of the big macromolecules that we're going to talk about. Our friend nitrogen. Yeah, nitrogen's going to make its appearance in 2 of them. We're going to see that in proteins and nucleic acids, which we'll talk about in a sec. And then phosphorus, you may see in, definitely see it in nucleic acids, and you will see it in some lipids, known as phospholipids. And those are those guys that kind of look like this when we drew them, that go on to make the lipid bilayer. But we're going to break all these down as we go. And, yeah, pretty much. We'll see them all as we go. Let's look. So organisms exchange matter with the environment to grow, reproduce, and maintain organization. What are the major macromolecules and the monomers? So if you said something like carbs, and the monomers there are going to be glucose, or if you would have said simple sugar, I'm okay with that. That works. Simple sugars, I guess, would be fine. If you said protein, and you said something along the lines of amino acids, I'm very happy. If you said nucleic acids, and a nucleic acid is things like DNA or RNA, and that is going to be made up of a nucleotide. And if you said And now this one's a little tricky because it's not technically a macromolecule, but it is made up of parts. I would like you to say glycerol and fatty acids. And again, this is the one they're not going to talk too much about. This one's kind of not really a true macromolecule in the sense, but it is made up of parts. So if you said something along the lines of that, you're good to go. So as our carbs, and when we talk about carbs, I may mention starch. I may mention, I cannot believe I didn't say simple sugar here. Oh, well, yeah, I guess there it is. I'll get rid of that. Cellulose, which is what the cell wall is made out of. Glycogen, which is how animals store excess glucose. Starch is how plants store it. So these guys are more storage. This is storage, and this is going to be structural. Proteins are composed of monomers called amino acids. Proteins are going to go on to make things like enzymes. They're going to go on to make receptors. They're going to go on to be structural, which you may see in your muscles. They may go on to make hormones. So we're going to talk a lot more about these as we go. Catalysts, you may see that. Nucleic acids are going to be our friends DNA and RNA, which we kind of mentioned already. And lipids, of course, are going to be waxes, oils, fats. You may see them in your plasma membrane, so we're going to see a lot of those guys. All right, so let's see. And this is a trick, and sometimes they'll ask you these really simple questions, or they'll kind of embed it in a multiple choice if we were having any, and it's all about the giveaways. So if they're talking about a carb, carbs only have the big three, C, H, O. They only got the big three. Now, proteins add a big four, I guess, if you want, nitrogen. And then there are a couple of amino acids that have sulfur in them. I believe it's cysteine and methionine have sulfur in them. So when you talk about a protein that's made up of amino acids, if you look for sulfur, you may find it in very small quantities. A nucleic acid has, I guess, the big four too, carbon, hydrogen, oxygen, and nitrogen. And they have a phosphate group, so they're going to have a phosphorus. But proteins do not have phosphorus. So this is a really good way to differentiate. So nucleic acids do not have sulfur, and proteins do not have phosphorus. So that's a good way to tell. We'll see it more later when we talk about how they knew what genetic material was. And then lipids are typically going to contain carbon, hydrogen, and oxygen, unless you are a specific phospholipid, but they'll go out of their way for that. So they're not going to try to trick you on that. And that's going to have phosphorus. So my little star there, sulfur and phosphorus are the giveaways about proteins versus nucleic acids. So just a little trick they might put in a question for you. This I found this kind of cool picture, and it basically just brings it together, I guess. The big three, which are these three, they're found in hydrocarbons and carbohydrates and lipids. Then you add your nitrogen, and that's going to be in amino acids and proteins. And you add your sulfur, and then that could also be in some amino acids and proteins, but only a couple. And then you add your phosphorus, and that's going to be in DNA and in RNA. So kind of cool. And then some of the things that they'll do, lipids, once again, are going to be for some storage energy. They're going to be found in the cell membrane, which gets its own chapter later. Proteins are unbelievably versatile. I gave you a list earlier. And then nucleic acids, when we get out to them, are going to be all about usually your genetic info. So things like what you pass on to the next generation. Carbs are going to be quick energy, and they may also be structural in the form of cellulose. So a nice little kind of review slide there. And the last part for this video, 1.3, we're going to get into some macromolecules and how they're made, and how is water used to make and to break molecules. So if I'm going to take, let's say, an amino acid, and I'm going to add that to another amino acid, you're going to see later how this works, but they just can't stick together like Velcro. So what's going to happen is for them to form a bond, which we're going to talk about in a little bit, it's called a peptide bond, you have to release a water. So that's known as dehydration synthesis. Well, the exact opposite happens if I have, let's say, a little dipeptide, or I have two molecules, and I want to break them. I have to add that water back, and that's known as hydrolysis. So they are, in essence, opposites of each other, but we'll see that in a second. So here it is. When water is added to a molecule, it's going to, the H and the O split and join the monomers. And this is going to break them up. Or the word they use is cleave them. And that's known as hydrolysis. And again, I'll show you some pictures in a sec. When two monomers join, they need to create a bond. Well, they can't just make a bond out of nothing. They have to get rid of a molecule, and they're going to lose a water. And that loss of water is known as dehydration synthesis. And we will see that a lot coming up in the next couple of minutes. What you ultimately create is what I drew before, a covalent bond. It's not ionic. It's not a hydrogen bond. It's a covalent bond where electrons are being shared. So in this case, you have a polysaccharide. In this case, there are 5 monomers to that. And they're held together by covalent bonds right there. Those are covalent bonds. And if you want to break up that polysaccharide or that larger molecule, let's just say that this is starch. Let's just say. And let's say that these are all simple sugars like glucose. So you got 5 simple sugars, which make one giant molecule known as a starch or a polysaccharide. Well, by adding waters, hydrolysis means you add water. That's how you break them up. And now these monosaccharides or these simple sugars can be used later in maybe cell respiration, which again we'll talk about in Unit 3. Well, the exact opposite is dehydration synthesis. Oh wait, here's one more hydrolysis. This is like the chemical end of it. So you see here how this oxygen right here, this oxygen has the ability to bind to 2 things. It's binding to that carbon right there, and that oxygen is binding to that carbon right there. And that's a very, very happy disaccharide. Very happy. Loves life. Sucrose. It's glucose and a fructose. So that's a glucose molecule right there. There's a fructose molecule right there. Everybody's happy. It's great. But if I want to break that up, I need the help of an enzyme, which we talked about. In this case, the enzyme is sucrase because you're breaking up sucrose. And there's your water you have to add. And the water, what's going to happen is it's going to split up. And the OH group's going to go one way, forming a bond right there, which everybody's happy. The oxygen is going to join the hydrogen is going to join the oxygen that was already there. And you have just now made 2 simple sugars that can go their separate ways. You broke a bond using water. Well, dehydration synthesis is the exact opposite. Let's say that this, again, is a simple sugar, and this is a simple sugar. Remember the H2O you put in? Well, you're going to take it out. So if you look right there, you got 2 H's and an O. That's a water. There it goes. Bye-bye. Say goodbye to the water. And there you just made a bond. So you took 2 simple sugars and you made yourself a disaccharide. You built a bond, but you had to get rid of the water. Dehydration synthesis. Here it is again. And I don't really care what this is. It's a short polymer. It doesn't matter. But I want to add one. Well, the only way to add one is to get rid of something. And I got to get rid of the water because there's my new bond right there I just made. Because again, I just can't make a bond out of nothing. And there goes the water. So technically, to make this larger polymer, I needed to get rid of 1 water, 2 waters, and 3 waters. Oh, always remember that there will be 1 less water than there are monomers. So if there's 4 monomers, 1, 2, 3, 4, that means that you needed 3 waters to make that, and you would need 3 waters to break it. And this finishes up with a dehydration synthesis and hydrolysis video. This is a shorty, but hopefully it brings everything to light. So section 1.1 through 1.3 are done. I'm going to post this probably tomorrow early, and your next group is due on Sunday, and you'll be able to work on that then. Okay? So good luck, and hopefully you're taking this seriously.